Hello, I'm Benjamin Miss, and today we're going to be talking about the temporal lobe and hearing. The associated readings that go well with this are going to come from Brain Facts and from the OpenStax Anatomy and Physiology textbook. And I've selected certain parts of those readings that I think best fit what we're doing. And we're going to talk about receptors, hearing, and the temporal lobe in terms of how it's processing. They won't go into huge depth in the lobe itself. The focus will be on the sensation of hearing. And first, let's overview what the senses are and how these things work. So let's look at senses, right? So we talked about vision already, and we have a lot of very clear visual pathways, a lot of very clear studies on vision and how vision works. So we have a lot of other senses, and we already saw that, we've got these 5 senses that are kind of a meaningless statement. It's just a nice way to group things together, but it certainly misses out on things too. Remember, vision we could split up into our conscious perception of vision. I say, what is this? What color is this? And motion perception, which we saw is a separate neurological pathway where we're able to detect movement, even if we can't use that pathway to understand it. The basic senses are often described as the 5 that we know, plus the vestibular sense. I've heard it very kind of commonly described as balance. And that's an interesting sense. It's an interesting way of thinking about things. We sometimes talk about balance when we talk about movement. But the vestibular sense is sending a lot of information not to the temporal lobe, but to the cerebellum. We're still going to talk about it in this, section, because very clearly they come from the same organ in a way of perception. Well, in addition to those, we can break them down even more, and we consider that touch includes separate things. Pain, itch, and tickle are different. Deep pressure and vibration are different types of receptors. Balance and proprioception are often considered to be part of our sense of touch. Proprioception is where your body is in space, and in the rare cases where somebody loses proprioception, we can very clearly see that their entire experience of the world changes to the point where they cannot no longer move as they did. Smell and taste also can be broken down, where their sense of smell includes 2 different cranial nerves and their involvement. We think of olfactory in terms of when you smell something, we tend to think of just our olfactory nerve and general olfaction, but we also have this other component, which is almost like a chemical part, the trigeminal nerve, telling us more about how it smells in terms of, chemical type senses, hot, cold, cleaning products, burning. Taste, we already know we have a number of different receptors, and we also have temperature and texture that are part of taste. Sometimes you don't like something simply because of how it feels in your mouth. Well, we talked about photoreceptors when we talked about vision, because vision is very heavily involved with light. And so we have very specific receptors that deal with light. We also have mechanoreceptors, chemoreceptors, and thermoreceptors involved in touch and smell and taste. Well, mechanoreceptors are a mechanical process. They mean, for instance, you want to feel something, you press down, and you are causing a physiological change in the receptor itself that then leads to the signal. That's the mechanical receptor. All right. The chemical receptors, chemoreceptors, for mostly our sense of taste and smell, organic type molecules, then things like our sense of bitter and taste. Thermoreceptors are about temperature. And so we absolutely have this ability in our skin to detect the temperature. And yet even then, we don't always, that's still not everything. We think about what we're not able to detect. For instance, some species have receptors for magnetic fields. right? Birds and sharks are able to detect magnetic fields. They use them in movement. There is a fish that can detect electrical fields and has a massive brain dedicated to using electrical fields to find prey in murky water. We have snakes that can detect temperature in terms of infrared. So we know that there's this wide range of abilities. We also have other receptors that we rarely talk about, but these are part of our sensation, often automatic, right? Deeper levels of processing that just kind of give us information. You drink when you're thirsty, right? But you're thirsty because of osmoreceptors that sit around, in some cases, our ventricles telling us, hey, has the water level dropped? You know, is there less water pressure? And should we have something to drink? Now I think I just drank my tea because that was a dry mouth receptor. Baroreceptors or blood pressure, we have those. The carotid sinus is part of our cranial nerve. And then we also have the aortic arch giving information on our vagus nerve. Our heart giving us information on blood pressure and what we should do. Now let's look at vision and hearing, right? These are what we really consider our main senses. And we think of this in terms of which one is faster, what do we process first? Well, most people, interestingly, would say that we process light faster because sound travels at 340 meters per second. Light travels at about 300,000 meters per second. That is like 1,000 times faster, if I'm doing the math right on that. Extremely rapid. So we also consider what this means in terms of perception, because our intuition here may not be correct. Let's do an experiment. What I want you to do is I'm going to play some sounds, and I want you to look at my lips while I'm playing these sounds. Are you ready? Ma, 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 ma. Now, what you heard, what you perceived was me doing this and this. Now, the first one is related to the sound ba, and the second one is related to the sound fa. Okay, so far, so good. The issue is I only played the sound ba. If you'd like to go back and listen, close your eyes and play that part of this again. Now, this is called the McGurk effect. It's a powerful effect, and it actually shows us how our mind processes things in an interesting way. So, you are using visual information to change your sound perception, but it's even cooler than that. Even though light travels faster, our auditory perception is nearly 4 times as fast. What's going on? In fact, the only way for you to get light first, right, in terms of processing, is if someone's shouting from like half a mile away. You know, it might even be more than that. I haven't done the math recently, but it's really far away. You know, you've heard of cases where, you know, on the ancient battlefield, someone would see gunpowder. You would see the gunpowder rise up, you know, or the smoke, right, from the rifle before they, you know, heard the blast, right? But you have to be very far away for that. And then you have the cases of if you're waiting, if you're a runner and you're at the starting line and somebody has the starter pistol and they're going to fire the gun, should you look at the gun or listen for the sound? And people say, well, light travels first. You should look at the gun and look for the smoke, right? No. Sound is processed first. So even though light travels faster, 1,000 times faster, you process sound faster. Which means we are going to actually hear, right? We are going to actually hear the sound first. McGurk effect. You're watching my mouth. Your brain processes ba, waits for the information from my mouth, and then says, oh, no, that wasn't ba, that was fa, right? You hold the information, and you wait for it all to come in. what's going on, right? What in the world is our mind doing? It's changing itself. It's changing our perception based on this, and it means it's defaulting to vision. When somebody's talking in a loud environment, you look at their mouth. It gives you a lot of information. It's useful. There's nothing wrong there, but it also tells us that this is what's happening. Now, the reason for this is surprising, right? Because you're like, why? Why are we doing this? Well, most of our pathways, our audio and visual pathways, right? Our auditory and visual pathways are ionotropic receptors. Remember back, we talked about the synapses. Two types of synapses, right? Metabotropic and ionotropic. Our metabotropic synapses are like the Rube Goldberg machine or the OK Go video, where it's like this causes this, causes this. Ionotropic is open an ion channel. Well, mechanoreceptors, like those in our inner ear, are mechanical receptors, right? Mechanoreceptors. They open a synapse. They open something. They open a door. Most of our synapses between those are also mechanoreceptors, right? So all of our visual audio pathways receptors are mechanical. You have about the same number of synapses in your audio and visual pathways, just about. But in your visual pathway, that very first one, when light hits your eye, it is a metabotropic receptor, which means in order to convert your retinol into something that can cause a signal, that can send a signal that says, hey, a photon just hit here, you have a metabotropic process, which takes time. And metabotropic processes are so slow, comparably. We're talking milliseconds is all it takes for your, the signal to get to your, auditory cortex. But we're talking about metabotropic receptors being very slow, only in comparison. Again, we never think about the fact that, we're actually holding information, lining it up. It all seems to happen instantaneous for us. And yet it was Wilhelm Wundt, in the late 1800s who actually started to time these things and was like, these processes are instantaneous. It takes some time, right? And we're looking at our wavelengths here on this image, which tells us, when we're looking at visible light compared to radio, and then we even go these big auditory waves, how far apart these things are in comparison. Now I got another experiment for you. I want you to listen to this sound here. Tell me what's happening. Well, it should sound, it stopped. It should sound like a falling pitch. And we wait and we listen for when does it stop? How long can you make a note go lower? Sounds eerie, doesn't it? Well, these are called shepherd tones, and they were discovered in the 1980s by Dan Deutsch, who was a UCSD professor. Basically, they're multiple parts, right? So it's basically a sound made up of 8 sounds. So each note is 8 octaves worth of sounds, right? And so we kind of average them and hear them all together. So as they rise, in this example they rise, but if you flip them, they're falling. Basically, the only thing that's changing is you get to the top here, this top note jumps down to here to continue to rise. Everything else is still rising. So we hear this overall rise that seems to go on indefinitely. Now listen to these sounds and tell me which one is the second sound, higher or lower? Are we ascending or descending compared to the first? So, was the second sound higher or lower? Okay. You hear this, and if we listen to it over and over again, you hear the same thing, but we interpret them differently. So different people will interpret these things as rising or falling based on their own perception. When we think about things like that are ambiguous stimuli lead to these odd kind of perceptual things. What was it, the green or the blue or gold dress, the Yanni or Laurel sounds. These are those ambiguous stimuli that give us quite an interesting window into the mind. These tritones, Deutsch's tritones, were constructed as the first tone and the second tone were always a half octave away, so the distance between them could easily be interpreted as up or down. Now let's consider how we process pure tones, voices, music, and noise. What are pure tones? These are only produced mechanically often using a computer. I think MIDI files were like this, right? Where it's just kind of like a pure sound, a single sine wave, right? Most sounds that we hear are a combination of things. How complex are they? And how much of our cortex is processing each of these things? So think about what this means. How much cortex are you using for pure tones, voices, music, and noise? Well, when we think about it, most people would say, okay, the higher ones, the more processing is going to be voices and music. The lower ones are going to be pure tones and noise. Well, what is it about? And it is exactly true. Where when we look at our vocal sounds, we clearly see that we have these, different areas of the brain involved. And so we see that as we kind of go down this from the top to the bottom, a lot of processing is with vocal sounds. Here's a summary of these things we saw, right? The pure tones only use the auditory cortex. And there's a terminology thing here we need to pay attention to. The auditory cortex is also called HG in a lot of written kind of articles and places. And this is from Heschel's gyrus. It's an old term that is still used sometimes. We generally call it the auditory cortex, or A1, but we will sometimes see HG. That's what HG means. In the same way we talked about the striate cortex, we talked about the visual system, the striate cortex being the occipital lobe, HG being the temporal, well, specifically the auditory cortex in the temporal lobe. Now noise gives us this, and also we call it the STG, the superior temporal gyrus. So a little more than simply our pure tones. And then the frontal lobe motor areas as we try to figure out what the sound is and what's causing it. Music gives us these, but also sensory and Broca. So there's more involvement. And Broca's area is in the frontal lobe, involved in speech. And then we go to vocal sounds, and then we get a lot of the STG and Broca's area. Because now there's so much information in speech, and that's what it's about. There's information in music, and if you're trained musically in any way, you understand that music has phrase structure in a similar way to speech, and yet it doesn't have quite as much information. While it can evoke emotion, it could be a beautiful experience, it really not, kind of your bang for your buck is a lot higher if you want to evoke emotion through lyrically, generally, poetry. That's going to give us a bigger effect. Light is processed slower than sound because, and it's got to be right, red or blue, and the answer is vision has a metabotropic receptor. Thermoreceptors detect what? Sound, movement, temperature, or water balance? And the answer is temperature. Osmoreceptors detect. Same options. What do you have here? Sound, movement, temperature, or water balance? And the answer is water balance. Hearing. We got the outer, middle, and inner ear. We're going to go through each of these components, and so let's take a look at what they are. All right, and we'll see the same image on our following slides. Starting with the outer ear. The outer ear is interesting. It's called the pinna, sometimes the auricle, if we're looking at our anatomical texts, and the ear canal. So everything on the outside up to the tympanic membrane, not including. Now what's interesting about the pinna is it actually does something for us. And if you want to kind of see, if you use your hands to kind of give yourself big animal ears while you listen, you can hear a difference. And if you were to pin your ears back, you can too. So clearly we're using this fleshy structure here to direct the signal, right, the sound. But it's not nearly as interesting as the other processes involved. So the middle ear contains a tympanic membrane, also called the eardrum. If you've ever seen a timpani drum, that's kind of what they call it, right? It's a drum. And then you've got the malleus, incus, and stapes. These are also called the hammer, anvil, and stirrups. These are the three smallest bones in the human body. You have two sets of them, right? And they are tiny. But they are bones, and they help to attenuate the signal between the tympanic membrane and the oval window of your cochlea. So this middle ear includes from the tympanic membrane to the connection at your oval window, and then the round window at the bottom. You see that a little lower down. And so what's happening is the oval window is where you have this contact between the stapes and your inner ear. Now, this is where you've got fluid in your inner ear, inside your cochlea. And what you have to have is movement, which means your round window has to move. So these are all vibrations. It's all vibrations. Sound is all vibrations. And so these ossicles, they're called ossicles, the malleus, incus, and stapes, they're rapidly vibrating, and the signal is hitting your, getting to your cochlea, where it travels through that, that snail-shaped piece, and is then going to affect your these hair cells that are your auditory receptors. Now you've got these three fluid-filled tunnels in the cochlea itself. So we're going to look at how this works. So the inner ear is where we transduce these sound waves into something that our brain can process. So here is producing the sound, and I suppose a tuning fork is another example of like a pure tone, right? A single frequency. And you have this signal that's traveling through your ear canal to your tympanic membrane. Well, in your tympanic membrane, it is then going to transfer to your malleus, incus, and stapes, which will then transfer to your cochlea. And so you will cause this bending of the membrane, causing the hair cells to vibrate, and then that signal is saying where they are. And we're going to understand how this works in terms of frequency and pitch and sound. Consider. We take a cross-section of the cochlea, and the cochlea is going to have these three fluid-filled canals, and in them, in the middle, you have this organ of Corti. And the organ of Corti has these little hair cells that are going to be connected to your auditory nerve. So the outputs from these hair cells are going to send a signal that's going to eventually get to your brain. The sound, how far it gets, is going to be based on this. So we're thinking about, here's this sound wave, how in the world are we pulling it apart? And I want to say this because when it comes to auditory perception, people who research this stuff get really excited. And the reason for that excitement is your auditory system does something that's really hard for a computer to do. And it took a long time for computers to basically have the kind of processing power to do this that we do instantly and effortlessly. Which is to say, okay, you've got all these sound waves. You've got these components of the sound wave. When I speak, it's not a pure tone. There's a number of different frequencies in my voice, and in any voice, and in musical instruments. It's timbre. It's a combination of these sounds. And so these frequencies are being basically chopped up and then put back together. And you're taking this massively complex wave and pulling out these tones to put together in order to figure out what's being said. If you were to take just the sounds themselves, the phonemes, the letters that we have in our language, like sh, f, t, you're pulling these things out and putting them back together into speech, which is a fascinating task. Well, the parts of the sound wave, and how far you travel is based on those frequencies. So perception of sound is based on a couple key components. So you've got amplitude, right, which is a physical intensity the height of the wave. The louder the sound, the higher the amplitude. The frequency refers to the number of compressions per second. So loudness is our interpretation, our perception of amplitude. Pitch is our perception of frequency. And amplitude high versus low works for volume high versus low. And frequency high versus low works the same way. A high frequency sound, your sound waves are closer together, sounds higher. Low frequency, the sound waves are further apart, sounds lower. Lower frequency sounds tend to travel farther if they're at the same loudness, right? Same amplitude. And the reason for that is these sound waves are going to peter out over time. They're going to lose power. Well, a lower sound can travel farther because it doesn't have to do as much of this, right? It doesn't have to do as much of up and down. It's just this longer wave, which means it doesn't travel as far. So you travel farther if you have less movement. And so we see here, amplitude, louder is higher. And then for pitch, the more waves you pack in, the higher you are. If you speed up a video, the sound gets higher because you're packing it in. The Doppler effect, if you've ever listened to an ambulance come toward you and then past you, right? It sounds higher pitched as it comes toward you and lower pitched as it goes away. So as it comes towards you, it's compressing the wave, right? This sound is going, you know, you're getting the sound wave as the thing moves in this way, and as it moves away, right, the sound wave is being pulled a little further apart. It's a slight difference, but it's enough for us to perceive. Now, we also have in our perceptual system what is called a tonotopic map. We have this both in our cochlea and in our temporal lobe. Your brain has this too. Now, we also look at what happens when we age, right? How do sounds work? Well, you will notice that at the base of the cochlea, you have this 20,000 hertz. Very few can hear 20,000 is a very high-pitched sound, right? And at the very base, you can get down to 250, 20 hertz, very low-pitched sounds. Well, when you hear loud things, how do you get deeper in, right? Well, the answer is lower pitches tend to travel deeper in. Higher pitches don't. And because of the movement, you get more damage at the entry here. So as you age, you lose the high-pitched sounds. You lose the high-frequency sounds, right? Which is why we see that older people have a harder time hearing those things. There was the ringtone called the mosquito that people in often middle school would have on their phones, because you could hear your phone and your teacher couldn't, because it would be basically at this, maybe 18,000 hertz, which was too high for older people to hear. The bad news is we do a really good job of damaging our hearing. We have so much noise in our environment. And I can't tell you how many times I can hear somebody, in my car, and I can hear somebody's music in the next car, and I have my windows up, and they have their windows up, and they're just destroying hair cells, these little cilia that don't grow back. So you do damage here, you're not going to get it back, and you're going to lose those sounds. And there's a lot of issues that come with that, because it's, the more you lose, the harder it is to distinguish, right? People age, and it's harder to hear voices in crowded rooms, for instance. It's harder to distinguish things. There's ways to fix this. With voices, one-on-one, even someone who's got a lot of damage can often hear that, because voices tend to be in this range here, toward the middle and toward the back, which is nice. So no matter what, and it's a logarithmic scale, which means you've got a lot more space for your lower tones, and then high tones are compressed, which again should make sense, because in nature there's not that many higher tones, and our auditory systems should be well tuned for voices, and voices are going to be in this nice middle range here that you really have to, you're going to retain. Right. Now you're also going to see damage too, and things do change over time. So what we see here is this. Now let's go back. Let's look at our, we're looking at our tonotopic map here. if you lose this, if someone has hearing loss, one of the ways that you can help them, hearing aids, can actually take, some hearing aids are just going to boost the volume. But you can also say somebody has hearing loss here, and what they will do is they will take and say, okay, your cochlea is only working from 5,000 hertz on back. So they'll take this space here and remap it. So you'll shift everything a little further back because this part's not doing anything. They won't be as sensitive. And again, hard to hear in crowds, but they'll be able to hear those sounds. Now what's interesting is this tonotypic map up in your cochlea translates to your auditory cortex. So you see here these sounds are these sounds. The lower sounds are in the front, toward the front of your brain, right? The anterior, and the posterior have the higher pitched sounds. Now this means you have cortex dedicated to these higher pitched sounds, which is a problem. Because if you lose this, you've got this brain tissue that's not doing anything. And in a way, tinnitus, some forms of tinnitus can be similar to phantom limb because you've got a sensory organ that no longer has the ability to sense. But your brain still has all this dedicated stuff to these high-pitched sounds. So you can have this experience. Because your brain's not going to say, oh, we're not getting any inputs anymore here, let's just not use this. It's going to use it for something else. And you're going to perceive that as sound. What is a term we use to describe pitch? Amplitude, frequency, or loudness? And the answer is frequency. As you age, you first lose the ability to what? Loud, mid, low, or high? And the answer is high frequencies. The higher frequencies are processed where in the auditory cortex? Now we go back to our location terms. Dorsal, ventral, anterior, or posterior? Where are the higher frequencies processed? And these were posterior. Higher frequencies in the back, lower frequencies toward the front. This is in the inner ear. Only one of these things is technically the inner ear. Pinna, tympanic membrane, cochlea, oval window, and the answer is cochlea. Now let's take a look at this image here. Can you identify these pieces? Right. So it's fairly non-abstract, right? You can see the pinna in the ear canal. This piece here at the end represents a tympanic membrane. Then you have your ossicles, malleus, incus, stapes, connecting to the oval window, and here's your round window. This is your eustachian tube. Now, if you've ever had tubes as a child, right, this is likely where they go because you get a lot of fluid buildup that can damage your hearing. It can cause permanent damage. You put tubes in, and it allows for that drainage. With modern medicine, we have avoided many things that our ancestors might have faced. Many of us would face hearing loss that was able to be corrected for. In the same way that they can test babies, they can test newborns for hearing, which is just a fascinating thing, right? They can often detect issues early on because what they do is they put these adorable little headphones on the baby, and then they detect back here on the brainstem, they're looking for, basically, they're using an EEG type system to detect signal. So if you're hearing it and you're producing a signal here, it means that this stuff's working, right? So they can actually, you can't ask a 2-day-old baby, okay, baby, I'm going to play this sound. Did you hear it? Raise your hand if you hear it. But you can detect if their processing is working. And here's our cochlea. And we see our semicircular canals here, which are not the same as, part of the cochlea. This is part of our vestibular sense. How we perceive pitch is interesting. We have a couple ways that this works. So first, place theory and then frequency theory. Place theory means that along your basilar membrane, right, along this this organ in your inner ear, as you get deeper and deeper in, you have high-pitched sounds at the end and low-pitched sounds inside. So basically, whatever hair is detecting that, you've got your sense. Frequency theory suggests that your auditory nerve axons produce action potentials at the same frequency as the sounds, which means if you have a 200 hertz sounds, you've got neurons that are firing 200 times per second. Well, this works to a point, right? But you can't fire 20,000 times per second or even 10,000 times. Your neurons would die. That's just too fast. Now, you can't get your sodium potassium pump, restabilizing at that point. Now here we have our the idea that each of these theories is helpful. Low frequency sounds are best explained by frequency theory, and high frequency sounds are best explained by place theory. And again, these theories are explanations for how we perceive sounds. We basically have two mechanisms by which this works. But we also have something called the volley principle, which is for sounds just about in the voice range, which is, kind of you want to have like extra help, right? Because voices are extremely important for us to perceive. So you've got these volleys of impulses. In a similar way to like a piston, like pistons in an engine, if you have like an 8-cylinder engine, you don't have 8 cylinders firing at the same time. You have them firing one after the other, but it allows you to fire very rapidly, right? One cylinder can only, fire up and down, up and down. 8 cylinders can all go up and down, but if you synchronize them, right, it means you can basically, create 8 times, the speed of movement along there, right? So you've got all these axons working together to give you these sounds. There are some things that can happen with sound too as we get deeper into perception. So there are some people who can't perceive music, right? They basically, you can call them tone deaf. They hear music, and the description is as if someone was banging pots and pans together, a very unpleasant sound, right? They tend to have some very specific changes neurologically, thicker than average auditory cortex in the right hemisphere, and fewer connections from the auditory cortex to the frontal cortex. And the frontal cortex being where we interpret music, right, structurally. Quite interesting. Then, on the other side of this, you have absolute pitch or perfect pitch. And in the same way that we think that people have the same abilities we do, which they don't, right? We think that if we perceive things a certain way, other people do too. There's a huge range between perfect pitch, where you can hear a door squeak and say, B flat, right? Or, you can't perceive it at all. This huge range of experience, right? There's a genetic predisposition for this. Training is required. And interestingly, people who come from cultures that speak tonal languages like Mandarin and Thai will actually have greater chance of having perfect pitch due to the necessary task of figuring out pitch in sounds. Well, we've made it through to the auditory cortex, A1, and we see it here in yellow. We see STG, right, in the same way we like to use our locative terms. STG, we said, is a superior temporal gyrus. Top of the temporal lobe, superior, and then the gyrus being the raised area. And it's kind of that part that folds in, so if you separate the temporal lobe from the parietal and frontal areas, you can kind of get, there's some more tissue in there. Now, I've seen a weird myth here where people are like, oh yes, the ears don't cross, like your ear information goes straight to your brain, which no, because your sinuses, where all these things are embedded, are here. Your brain's up here, right? This is sinuses and embedded stuff. And you feel your skull, right? So your ear is here. It's outside of that kind of hard part of your skull in the spongy sinuses. Most of your ear stuff is crossing to the opposite hemisphere in the same way everything else is. But in a similar way to, as we detect the location of sounds, we're splitting them up in a visual world in that way. Now here's the pathway. All right, so this is our deep dive into where this information is going. You've got your cochlea, and your cochlea is sending information to your pons, basically in your brainstem. You have your superior olivary nucleus that is now sending information up to your, you see it crossing here from one side to the other, and this is sending information to your inferior colliculus. Your inferior colliculus is in your midbrain. It's right next to your superior colliculus that does vision. So part of our visual system, specifically for visual attention, something that draws your attention, your inferior colliculus is involved here, right? So what is important for you to pay attention to is directly part of your auditory pathway. Then you go to your MGN, your medial geniculate nucleus, which is, interestingly again, right next to your lateral geniculate nucleus that does vision. Vision and hearing right next to each other in the brain in more ways than one. Inferior colliculus for auditory right next to your superior colliculus for vision. Medial geniculate nucleus right next to your lateral geniculate nucleus. Medial does auditory, lateral does vision. And then you send the signal to your auditory cortex, and you see that area highlighted here. In the auditory cortex, outside of A1, you have more things happening. So you've got more processing here. You see your auditory association cortex in that kind of lighter yellow. And you've got kind of more complex things, auditory objects. And we see that we tend to see a lot of areas where we see changes over prolonged sounds. In the same way, our visual system really likes change. It doesn't like static. You stare at something, you get this kind of hallucination. Hearing problems. I mentioned you have different things you can do for kind of hearing aids, but some of them won't work at all. If you have conductive or middle ear deafness, or even some issues with your cochlea, you can often have some kind of hearing aid to deal with it. Nerve deafness is where you just have no signal coming from your cochlea to your auditory nerve. And if you're familiar with cochlear implants, so basically a non-functioning cochlea, right, your inner ear isn't working, they can implant what they call a cochlear implant, and this will send a signal directly to the auditory nerve in order to make it possible for you to hear. So there is, in many cases, some kind of solution for deafness if you want to, maintain this hearing route. Just like with people with visual issues, however, if you have deafness, the parts of your brain associated with hearing will then be used for something else. Just like if you're born blind, the parts of your brain that do vision will start to do other things. They'll still maybe help you navigate, get through space, create maps in the same way that we would do things as visual. And I mentioned tinnitus already as problems with, your damage to your cochlea. We also have interesting ways for localizing sounds. Newborns can generally know which side the sound is on, left or right. This is a very old, very deep ability that we have. We will basically be able to calculate rapidly in our auditory system the responses of the two ears and differences between them. And these three cues we use are sound shadow, time of arrival, and phase difference. So our low frequencies, we tend to use phase, and high frequencies, loudness differences. And so here's what we mean by these things. So the time of arrival is a difference in time of arrival at two ears. So if a sound's coming from over here, this ear is going to get it first. And so because this ear gets it first, after this one's going to be like, okay, it's coming from over here, right? It's on this side. Sound shadow is produced by the head for frequencies from about 3,000 to 4,000 hertz. Basically, your head creates a shadow as the sound's coming this way, and so there's less volume here because of my head being in the way. And then we can also use what we call phase difference, which is, remember we got our sound waves that look like this. A slight shift in sound wave where this ear is getting it starting here and this ear is getting it starting here. So you basically have these slight differences in sound phase. And the phase is where you're starting in that curve of frequency. That's a tough one to understand. So again, we think about our sound wave. Where is this ear getting the wave when this ear is getting the wave? So if the sound's coming from over here, it's a low sound, this ear is going to get the wave. It's going to appear to be about the same time, but the wave is going to be in a slightly different place when it hits this ear than when it is perceived by this ear. People with this have impaired ability to detect pitch and pitch changes. What is this called? Medial geniculate nucleus, absolute pitch, amusia, and sound shadow, and the answer is amusia. Medial nucleus can't be right because we all have that, and that's part of our auditory pathway. The higher frequencies are processed where? In the auditory cortex, dorsal, ventral, anterior, or posterior, and the answer is posterior, in the back. If the round window does not expand to allow for fluid movement in the inner ear, a person would probably have, is it conductive middle ear deafness, nerve or inner ear deafness, amusia, or no auditory problems? And the answer is conductive or middle ear deafness. Now let's consider something that's not quite temporal lobe, but it travels on the same pathway, right? So the vestibular system is right next to the cochlea, and it uses the same nerve. It directs compensatory movements of the eye and helps maintain balance. You turn your head, you know. And the reason you know is the 2 otolith organs and the 3 semicircular canals. The otolith organs are these, the saccule and the utricle. And these are part of the cochlea. You know how flashbangs work? Like the SWAT team has this thing. It's just this bright light and loud sound. And it not only blinds and deafens you, but it also can hurt your balance. Basically, it can knock you down and you can't stand up because it's so concussive. It can move around the fluids and the little otolith, the little crystals, these things in these organs. And so your brain doesn't know which way is up. And if your brain doesn't know which way is up, you have a hard time standing because you're not going to have that kind of balance. And so these are going to send information along the same way, but it's going to go to your brainstem and cerebellum generally in order to help you do these things. For the temporal lobe, there's a few things I want to mention before we finish. The temporal lobe has our what pathway. So you've got from your visual system going up, it's where, and going down in the temporal lobe, it's what, right? So again, identification, semantic, information, meaning. We also have the fusiform face area, which is on the bottom of our temporal lobe, which gives us information on faces. It's putting together things, putting together all this visual information to say, boom, that's my friend, or that's someone I don't know, or that's a person that I've seen, right? We recognize faces in our temporal lobe. We have this structure. And then another big thing that our temporal lobe is doing is memory. And the reason for that is our hippocampus is in our temporal lobe and surrounded by these medial temporal lobe structures that help us to do memory. And we're going to dive much deeper into memory as we go on and how memory works. And so part of memory is temporal, but not all of it. So memory for information about ourselves and information is there, but there's other types of memory that we're going to have to consider. And then language recognition is generally happening in our temporal lobe, understanding, right, these components. And so language is also going to take up a great deal of our discussion too. We're going to spend a lot of time because language isn't just temporal, and there's a lot of processes involved neurologically. And so that's our temporal lobe and auditory stuff for today. Thank you very much.